Good evening, everyone. Hello, good evening, everyone. Hello, very good evening to all of you. I hope you can hear my voice clearly. So, can I have some quick comments, please? If you can hear voice clearly and if I am audible, if I am visible, if you can see the screen. Hello, this is Vaishali Kamdar from Spark Academy. Very good evening to all of you. Warm welcome to all of you. And today we will be discussing about anatomy of flowering plants. We have discussed morphology yesterday and today our topic is anatomy of flowering plants. So am I audible to all of you? Okay, let's move towards the topic. Before starting the topic, I'll remind you all of you that you can follow us on sparkacademyonline.com and Instagram, Telegram, Facebook, whatever you want. You can follow Spark Academy in any of this link. Our today's topic is anatomy of flowering plants, so it is as equally important as morphology. So around 3 to 4 questions normally asked from this chapter. So structural organization of plants involve morphology, that means outer structure, on anatomy, that means internal structure of flowering plants. So 3 to 4 questions are mostly asked from this chapter. So around 12 marks, around 12 to 16 marks it will give you. So stay tuned with me. Next 55 minutes, stay tuned with me. We are going to solve a few PYQs and I'll be giving you few homework questions also. So those who have not joined, please join fast and ask your friend to join fast, because today we are going to, this is one short program to discuss anatomy of flowering plants. Before starting the chapter, uh, uh, let me show you that you can download this app from App Store or Play Store, that is Spark Academy app. So after downloading this app, you will be having you will be having access to free handwritten notes, study materials, PYQs, that is previous year questions, and free mock tests are also available on that. So let us start at chapter, that is anatomy of flowering plants. So here we are discussing about basic internal structure of plants. So you all know that cells together make tissues, tissues together make tissue system or organ, and organ system, and then an organism. But in case of plants there is no organization. There tissue, it is still tissue system only. These tissues, they will not further organize to form organ or organ system. Okay, so in case of plant anatomy it is very easy. We need to study till tissue system only. So tissues of plant, it is divided into tissues are divided into two main parts, that is meristematic tissue and permanent tissue. So meristematic tissue, that means continuously growing tissue, and permanent, that means when this meristematic tissue loses their capacity to divide. If they lose their capacity to divide and they mature and differentiate to form a permanent tissue. Okay, so it is these are cells involved in the permanent tissue. Cells involved in the permanent tissue, it is coming from meristematic tissue only, but they are differentiated and they lost their capacity to divide further. Okay, so they become permanent tissue. Meristematic tissue, again, it is divided into two types on the basis of origin, that is promeristem, primary meristem, secondary meristem, and on the basis of position there are three: apical, intercalary, and lateral meristem. Permanent tissue is further divided into simple tissue or complex tissue and special tissue. Okay, so simple tissue and complex tissue we need to discuss here in this chapter. Simple tissue, that means uh, cells which lost their capacity to divide further, and same kind of cells making a tissue, that is called simple tissue. Different type of a cell, together they function for one, they work for one function, like transport of water, like transport of food, then they are called complex tissue. So then parenchyma cells, collenchyma and sclerenchyma, they are involved in simple tissue, and complex tissue involves xylem and phloem. Special tissue involves lenticephorous tissue and glandular tissue. So this we will be not discussing in this chapter. Okay, now types of meristematic tissue. See, those who have joined, if you have doubt in between also, you can ask me anytime in your chat box. I'm continuously watching the box if you are there, and if you are answering, or if you have any doubt. So I'll be answering all your questions. Next 55 minutes I'm with you, and you concentrate on my words, so all your doubts will be cleared. Okay. Now, types of meristematic tissues. So location of meristematic tissue are found in those regions which need to grow continuously. For example, root tip and stem tip, and contain, so all the tips, they contain meristematic tissue. So first, so at the tip region, whatever meristem is present, that is known as apical meristem. So it is known as apical meristem. So they are primary meristem, that means they originate in the early stage of the life cycle of a plant. So that's why they are called primary meristem, and they are found at the growing tips of the roots and stem. The shoot apical is present at the tip of the shoot, and its active division result in the elongation of the stem and formation of new leaves. The root apical meristem helps in the root elongation. So basically they help in the root elongation. In case of root, there's a region of meristem, over that there is one cover which is known as root cap, which is protecting this particular tender root tip. Okay. So types of meristematic tissue. Intercalary meristem. So they are primary meristem. Intercalary meristem, they are also primary meristem, that means originated in early stage of the life cycle of the plant. So it is present between the masses of the mature tissue. Remember this point, question can be asked, which type of meristem is present between the mature tissue. So that is intercalary meristem, which is present between the mature tissue, or you can say differentiated tissue. Okay, it is present over here at the node region, at the on the basis of leaves and internodes. So it helps in the growth of the branches and the leaves. And very important point for NEET, these two are very important points. So regeneration of grasses after they grazed by herbivores, it is because of an intercalary meristem. Since intercalary meristem is an apical meristem appears early in the plant life, that's why they are called primary meristem. Primary meristem. Okay. Now types of, again third type of the meristematic tissue, that is lateral meristem. So lateral, it is responsible for lateral growth, as well as it appears late in the life cycle of the plant. So that's why it is a type of secondary tissue, secondary meristem. Secondary meristem. So lateral meristem, they occur in the mature region of the roots and shoots. They are called secondary meristem, and as they appear later in the plant's life, so it helps in the adding secondary tissue to the plant body and increasing the girth of the plant. So types of lateral meristem: fascicular cambium, interfascicular cambium, and cork cambium. There are three types of lateral meristem which appears in the late stage. That's why they are called secondary meristem, and that is responsible for It is present over here, which is responsible for increasing the girth. It is increasing in the girth of roots or stem. So meristematic tissue, you can see a quick revision of meristematic tissue. It is divided into three: apical, intercalary, and lateral. Present on the root tip and shoot tip. Intercalary present at the leaf base or the node, and lateral present on the sides of the stem and root. So apical responsible for the growth of the root and shoot, and intercalary responsible for the growth of the plant from leaf and nodes, and responsible lateral is responsible for increase in the diameter, that means increase in the girth. Next, meristematic tissue. Next, after that, it is next it is a complex, or you can say permanent tissue. In that, first it is parenchyma cells. First, look here. So first is parenchyma. So parenchyma tissue, first it is parenchyma. It's a type of permanent tissue. In that simple permanent tissue, so all the cells are of same kind. And this type of permanent tissue, it is having thin cell wall. Characteristics of it is thin cell wall made up of cellulose, and there is intercellular space present in between. Spaces are there, and intercellular spaces are there. Nucleus is also there, so they are living cells. Okay, if they contain chlorophyll in it, then they are called chlorenchyma, and if they have more air cavities, stored air, then it is responsible to give buoyancy to the aerial part of the plant, so that is known as aerenchyma. Next, collenchyma. So collenchyma is a strong flexible tissue that provides flexibility to the soft aerial tissue, and they are found below the epidermis in the leaf stalk. So what are the characteristics of collenchyma? You can see this type of angular deposition. Angular deposition or cell wall is uneven, thickened, and with the cellulose. Okay, and intracellular spaces are absent here. Unlike parenchyma cells, nucleus is present and tissue is living. That's why few chloroplasts may be present in the cells. Okay, collenchyma provides mechanical support to the growing part of the plant. So it is giving flexibility to the soft aerial part because of normal wind current or fluttering of the when leaves and all the aerial part of the plant, they flutter, they will not break off from the plant. So without breaking, they can bend without breaking. That is known as flexibility, which is provided by collenchyma. Then sclerenchyma is strengthening tissue. It is dead tissue, you can say. Nucleus is not there, and you can see evenly thickened with the lignin. Cells are even, you can see this type of thickening over here. They are evenly thickened with lignin. So lignin depositions are there. Lignin deposition is there. Intercellular spaces is absent. They are dead cells. Nucleus is absent here. Middle lamina of the wall between the adjacent cell is conspicuous because of the thickening of the cell wall. So this type of cell wall, it is also known as secondary cell wall. The sclerenchyma can be divided into two types: fiber with tapering ends, in which it is tapering narrow and tapering end, and sclerid, which is circular in nature. So sclerenchyma tissue is divided into fiber and sclerids. Most of the fruit wall and nut walls and seed coat of the some seeds like leguminous seeds, so they are made up of sclerenchyma. So fruit fruit pulp, seed coat, and nut wall. So they are made up of Sclerenchyma type of cell. Now complex permanent tissue. So complex tissue is made up of more than one type of cells that work together to perform a particular function, that is, they are known as complex tissue, like xylem and phloem. Xylem is a complex permanent tissue that conducts the water and mineral upward from the root to the plant. So this is also known as wood. Xylem, mean of xylo is wood. So this is also known as wood. Xylem is composed of 4 different kinds of elements: xylem tracheids, vessels, xylem fiber, and parenchyma. So then in xylem tracheids are long tubular dead cells with wide lumen and tapering ends. So look here. So these are tracheids with wide lumen and tapering end. Tapering, or you can say pointed end. So they have thick wall with the lignin. They have pores in their walls. They have this type of pores in their walls. Vessels is long cylindrical tube. These are vessels. They are long cylindrical tube-like structure that are placed one above the other at end to end. And they are also non-living with ligninified walls and generally possess pit. Vessels members are interconnected through the perforated in the common walls. The presence of vessels is characteristic feature of angiosperm. Function of these two tracheids and vessels both are main water transporting elements. Vessels are more efficient than tracheids. Xylem fiber. So they are long. See, this is xylem fiber. So they are long, non-living cells with thick lignin deposition. They have narrow lumen and tapering end. Remember, difference between fiber and tracheid is, in case of tracheid, it is having pores in their wall, and uh, lumen is wide as compared to the fiber. Then xylem parenchyma, they are living cells with cellulosic cell wall. So only one component or one element is living here, that is xylem parenchyma. Then phloem. Phloem is a complex permanent tissue that conducts food synthesis in the leaves of the different part of the plant body. Unlike xylem, conduction of the food occurs both in upwards and downward direction to storage organs and form storage organs. So phloem is in angiosperm is composed of sieve tube, while in case of gymnosperm, this sieve tube element is not there. They lack sieve tube. Instead, they have albuminous cells and sieve cells. They lack sieve tubes and companion cells. So this is also important point for exam point of view. So you need to remember this point, that what is the difference between phloem and xylem of gymnosperm and angiosperm. Gymnosperm lacks sieve tube and companion cell. Instead, they have albuminous cells and sieve cells. Okay, I hope there is no doubt in this 2 points. Now, phloem parenchyma. Phloem parenchyma cells are thin-walled and living. Phloem parenchyma is absent in most of the monocots. See, remember what is the difference between monocots and dicots. Now, you got the difference here. So that is parenchyma. Phloem parenchyma is absent in monocotyledon plant. Function, they help in the storage of food materials and other like resin, latex, and mucilage. All these type of secretions, or you can say the storage material, they are stored in phloem parenchyma. Phloem fiber, only non-living component of there are the only non-living component of the phloem. They have thick walled, elongated, unbranched, and like aphysis. So cell is narrow and lumen. At maturity, this fiber lose their protoplasm and become dead. So they become dead after that. So this fiber provides mechanical support to the tissue. Commercially, they are commercially what jute, hemp, and other fibers which we use. They are actually phloem fibers. The first formed primary phloem consists of the narrow sieve tube and it is also known as protophlloem. Later formed, it is known as metaphloem. Then tissue system. The tissue system involves three types of tissue system. Outermost skin of the plant, that is called epidermal tissue system. Okay, this layer you can say single layer. So that is dermal tissue, epidermal tissue. Then in innermost this ground, it is vascular tissue, and in between these two type, it is whatever cells in between it is filled up. So it is actually ground tissue. Epidermal tissue involves presence of lenticles, I mean presence of cuticle layer in case of stem. Okay, on I mean in case of all the aerial part of the plant, aerial part, but epidermal layer of root doesn't have, they do not have cuticle layer. Wait a second. They do not have cuticle layer. Roots do not have this. Okay, and epidermal layer also have stomata in them. Then ground tissue, mostly this ground tissue is it's composed of parenchyma type of cells. And vascular bundle, so it involves xylem and phloem. So vascular tissue system, the vascular system consists of vascular bundle, the phloem and xylem. If there is a cambium ring in between, it is present. If there is presence of the cambium ring in between these two, that makes it's open for the secondary growth. So secondary growth can occur in this type. So that type of vascular bundle, they are known as open vascular bundle. Closed vascular bundles like cambium, hence since they do not form secondary tissue, they are referred to as closed. So secondary growth do not occur in this type of closed vascular bundle, like in case of monocots, they have closed vascular bundles. So xylem and phloem can arrange in two different kinds of arrangement with the vascular bundles. So radial arrangement, when xylem and phloem within a vascular bundle are arranged in an alternate manner or different radius, you can say they are not in same radius, one after another. Okay, phloem, then xylem, like this, one after another. So alternate, they are arranged alternately. So this type of arrangement is called radial arrangement. Okay, this type of arrangement, they are common in roots. Conjoint, so when the xylem and phloem are situated at the same radius, look here, they are same, not in alternate manner. So same can be known as conjoint. In same way, so conjoint can be divided into two types, closed and open, on the basis of presence of cambium ring. Then anatomy of Dicot root. So we are starting now most important points, Dicot root. So outermost layer, let me explain you through diagram. Can you see here, this is outermost layer, outermost pink color layer, that is forming epidermis layer, which do not have cuticle layer. Cuticle layer is absent over here, and these cells protrude in the formation of unicellular root hair. Remember, root hair, they are always unicellular. So they are known as root hair. Then next region, it is cortex. This bigger region, all the layers, several layers, thin walled parenchyma cells, they are called cortex region. Then innermost layer of the cortex is known as endodermis. Okay, single layer of barrel shaped cells, they are single layer of the barrel shaped cells, they are called endodermis layer. Then pericycle, next to endodermis lie a few layer of the thick parenchyma cells known as pericycle. These are cells of pericycle, and this pericycle, it is responsible for initiation of lateral roots. See, in case of dicot, there will be like this: one is primary root, and from that lateral root system will be started. Okay, development of lateral rooting will be initiated, that is from the pericycle region. Then pith is small and inconspicuous. Pith is small and inconspicuous, and you can see only 2 to 4 patches of xylem and phloem in case of dicot root. 2 to 4 xylem and phloem patches or vascular bundle patches in case of dicot root. And these are also of open type. Because it is dicot root, it is open for secondary growth. Secondary growth can occur here. Okay, and there is presence of cambium ring in between. So these are characteristics of the anatomy of dicot root. Then monocot root. So outermost is just same like the dicot root. Outermost layer forming epidermal layer, it may protrude to form root hair, unicellular root hair again. Then epidermis, after epidermis layer, several layers of the cortex, the parenchyma cells. Then innermost layer of cortex is known as endodermis. Okay, tangential as well as radial walls of the endodermis cells, they have depositions of water impermeable waxy material called seborrheic. So they'll form Casparian strips. Pericycle. Monocot root do not undergo any secondary growth. So pericycle region is not responsible. Yes, Ramesh, there are JE crash course also. You can check out to Spark Store. I'll come to that point. You can check out Spark Store, or you can call us on these numbers which are flashing on your screen, scrolling on your screen. So monocot roots do not undergo any secondary growth. So pith is large and well developed. In vascular bundle, there are usually more than 6 xylem bundles in the monocot root. See, you can see this type of radial or sun-like arrangement. You can see this type of sun-like arrangement in case of monocot root. But in case of dicot root, it is just like a plus. Okay, so the sun-like arrangement, that means more than 2 to 4, more than 6, it is polyarch as a vascular bundle. And they are of closed type. Then dicot stem. In case of dicot stem, epidermis layer, and there is presence of cuticle layer in it. And if they protrude into the hair, they form trichome. The trichome, they are multicellular. Okay, they bear trichome which are multicellular. Remember, unlike root hair, these are multicellular with few stomata also. So cuticle layer is there. There is difference. So there is difference. In. Yes, Ramesh. I hope you got your answer. That crash course are available for JEE mains also. So then this cortex region, the cells arranged in the multiple layer between the epidermis and pericycle constitute the cortex. It consists of three subzones: hypodermis, cortical layer, and endodermis. Hypodermis, the outer hypodermis consists of few layers of collenchyma cells. Remember, this is collenchyma cells with angular deposition of pectocellulose, hemicellulose, and pectin. Then cortical layer, they are made up of parenchyma cells. Then endodermis layer, innermost layer of the cortex. It is rich in starch grains and referred as starch sheath. So pericycle, it is present on the inner side of the endodermis and above the phloem in the form of semilunar patches and sclerenchyma. Then vascular bundles, a large number of vascular bundles are arranged in a ring. The arrangement is characteristic of the dicot stem. If they are arranged in ring, then it is dicot stem. Then medullary rays, in between the vascular bundles, there are few layers of radially placed parenchyma cells, which constitute medullary rays. Then pith, a large number of rounded parenchyma cells with large intracellular spaces which occupy central portion of the stem constitute the pith. So pith is large here. Remember, pith is large here. Pericycle is made up of sclerenchyma cells. Vascular bundle, they are of conjoint open because it is dicot stem. Medullary rays, they are vascular bundle, they are few layers of radially placed parenchyma cells, and pith is large in case of dicot stem. Then monocot stem, one thing you can notice from the diagram only, that pith is absent over here. In case of monocot stem, pith is absent because of scattering of the vascular bundle throughout the stem. So that's why you can't see a proper pith over here. So in case of monocot stem, epidermis, it is outermost protective layer of the stem and covered with a thin layer of cuticle again. Cortex contain few layers of sclerenchyma cells. Then pericycle is present here. It is also made up of sclerenchyma, vascular bundle. Large number of scattered vascular bundles are present, and vascular bundles are conjoined, closed with peripheral vascular bundles, generally smaller and centrally located ones. Okay, you can see in diagram also, peripheral, these vascular bundles, they are smaller as compared to centrally located one, they are bigger. Phloem parenchyma, in case of monocot stem, phloem parenchyma is absent. Water containing cavities are present. So pith is absent as well in monocot stem. And dicot leaf, or you can say dorsiventral leaf. So dorsiventral leaf is found in case of dicotyledon plants, in which there are two surfaces, two epidermal surface, upper epidermal and lower epidermal. So upper surface is known as adaxial epidermis, lower surface is known as abaxial epidermis. So epidermis on the outside is covered with thick cuticle. So abaxial epidermis bear more stomata. See, that means lower epidermal layer. As you have studied from third standard, you are studying this thing. That is, lower surface of a leaf contains more stomata. Lower epidermis contains more stomata than the adaxial epidermis. Mesophilic cell. Mesophyll is the tissue of the leaf present between the abaxial and adaxial epidermis. So all these tissue, they are called mesophilic cell. Again, this mesophilic cell, they are divided into two types: palisade mesophyll and spongiform mesophyll. Palisade, that means they form this type of long oval. Okay, they are composed or tall and compactly placed cells with chloroplast in it. Okay, compactly placed cell with chloroplast. So they are tall, they are called palisade. And spongy parenchyma, they are oval and round shape, loosely arranged. You can see here. So they are loosely arranged. These are called spongy mesophilic cells. These cells functions for the photosynthesis because mesophyll contain chloroplast which perform functions of photosynthesis. Vascular system, the vascular bundles present in the leaves are conjoined and closed. There is no secondary growth occurring whether it is dicot or monocot leaf. Okay, so that's why they are conjoined and closed. So they are surrounded by the thick layer of bundle sheath. Thick layer of bundle sheath cells are there. Next, monocot leaf. So isobilateral leaf, that means both the surface. Okay, both the abaxial and adaxial epidermis bear same number of stomata. Same number of stomata. It is not like only lower surface has a higher number. And there is no differentiated palisade and spongy parenchyma mesophilic cell in case of monocot leaf. Bulliform cells you can see here. That is one of the most important characteristic of monocot leaf. That is presence of bulliform cells here. So this bulliform cells, which are modified adaxial epidermal cells. So they are present along with the veins. They are present along with the vein and which absorb water and make cell turgid. Okay, and when there is water stress, they become flaccid. First, there will be water loss from this bulliform cell. And they will become flaccid, and because of that, leaves curl inward to minimize water loss. So curling, important point to notice, curling of grass or curling inward of any type of monocot leaf, it is because of loss of water from the bulliform cells. Then parallel venation in monocot leaves is reflected in the near similar size of the vascular bundles as seen in the vertical section of the leaf. So vascular bundle, except in the main veins, they have nearly similar size of vascular bundles. Then secondary growth. The growth of the roots and stem in length with the help of the apical meristem is called primary growth, and apart from the primary growth, most dicot plants exhibit an increase in the girth. So this increase is called secondary growth. The tissue involved in the secondary growth are together known as lateral meristem. They are known as lateral meristem. So lateral meristem consists of vascular cambium and cord cambium. So now first, vascular cambium. See, remember here, vascular cambium can be divided into interfascicular cambium and fascicular cambium. See, in starting part, there will be development of the cambium ring like this in between patches only. Okay, in patches only like that. See, this is straight line I have drawn in between. They are fascicular cambium. So first it was developed in these patches. Then these patches are joined by interfascicular cambium formation. So because of that, a complete ring will be formed afterwards. So that ring is known as cambium ring. So look here. So in Dicot Stem, the cells of the cambium present between the primary xylem and primary phloem is intrafascicular cambium. Then interfascicular cambium is formed from the cells of the medullary rays adjoining the intrafascicular, sorry, here it is intrafascicular cambium as they become meristematic. So this result into formation of continuous ring which is known as cambium ring. Then spring wood and autumn wood. So activity of cambium is under the control of many physiological factors. Early developed in the spring season. It is known as spring wood or early wood. This cambium is very active and produces a large number of xylem elements. Large xylem, number of xylems are large in case of spring wood because water conduction is more as it is spring. Then in case of autumn, cambium is less active and forming fewer xylem elements, only narrow vessels. This wood is called autumn wood or late wood. Then heartwood and sapwood. So it is composed of dead elements with highly ligninified walls. It makes the plant hard, durable, resistant to attack of the microorganism. So it is darker in color and lighter color wood. It is known as sapwood. Involve the conduction of mineral and water. Water transportation is from sapwood only, not from the hardwood. Then quartz cambium, commonly known as phloem. Phloem, a secondary growth start due to the activity of vascular cambium and other cortical and epidermis layer breaks down. So they are replaced down to provide a new protective cell layer. Hence, sooner or later, another meristematic tissue called cambium phloem develops, usually in cortex region. So phloem is composed of thin-walled, narrow rectangular cells. Phloem cuts off the cells in the either side. See, it is actually converted into meristematic tissue, so it will transport newer cells towards the outside and towards the inside also. If it will transport it towards the outside, it will form cork, which is also known as phloem. Then the seborrheic deposits in the cell wall make it impervious for the water. The inner wall gives rise to secondary cortex formation that is known as phloem. So phloem, phloem and phloem together form one term that is known as periderm. So due to activity of cork cambium, pressure builds up on the remaining layers of the peripheral of the phloem and ultimately this layer die off and slog off to form bark. So bark is actually a non-technical term referred to the all tissue exterior to the vascular cambium. So bark is divided into two types, early and late bark. Then lenticels at certain regions, the phloem cut off closely arranged parenchyma cells on the outer side result into the instead of the cork cells. So these parenchyma cells soon rupture the epidermis cells. See, you can see this rupturing of the epidermis cells because of continuous addition of the newer cell towards the outside. This epidermis layer gets ruptured and there will be opening, lens type opening here. That is called lenticle, which will permit the exchange of the gases between the outer atmosphere to the internal tissue of the stem. It is found in most woody trees. So let me remind you all of you that this link of the Spark store is given in the description box. So you can buy anything, any course, long-term, short-term, crash course, full-length test, study materials, PYQ solutions of JEE Mains, NEET, MCET, IIT Foundation, IELTS, and all these courses are available in Spark store. So you can buy anything of that. So high quality courses with great offers and discounts are available right now. So just contact on the given number or you can simply visit to the Spark store. Link is given into the description box. So admissions are open and join. You can join now for hostel focused study hours, hostel plus focused study hours, that is long-term coaching. Offline coaching is also going on. Regular coaching are also going on 6 to 9 and 10 to 4 are the timing. We have our two branches, one at Mehdipatnam and one at Narayangura. Admissions are also open for online coaching. So NEET online coaching 2022, JEE Mains 2022, and MSET 2022 online coaching. So on daily basis, on demand, live sessions are going on right now. Just you are, what are you all waiting for? Just download, check out which course is specifically to you, which you want to join, and join right now because we are working on daily basis. Though their live classes are on demand, on student demand, we provide live sessions of particular um, what we say particular topic, and these all live sessions are recorded also. If you are unable to join at that time, then you can check these sessions afterwards, so you will not miss out any of the lecture which we will provide you. So that's the benefit you can get from this type of education, this type of academy. So what are you all waiting for? Right now, check the description box and check which course is feasible to you and call us on these numbers and join right now. So this is how on daily basis we are giving assignments and we are checking that assignments whether you have whether they have attempted, or test whether they have attempted or not. We can test we are we are conducting, and we can test analysis also doing. So this is our methodology, basic concepts. Normally we explain, then shortcut methods are also given. All model questions, exercise questions, PYQs, chapter-wise test, weekend test, grand test, test analysis. All these things are available. So now you can solve this question. It's question time. Question time, all of you. So now you can solve these questions. So first question is interfascicular cambium develops from the cells of what? Interfascicular cambium. That means cambium present between two vascular bundles. So this interfascicular cambium, it is developed from which region? Xylem parenchyma, endodermis, pericycle, or medullary rays? Come on, I'm waiting for your answers, all of you. First question, interfascicular cambium develops from the cells of which region? Yes, correct answer. Correct answer is medullary rays. So it is between, see, like that, these are patches, this type of patches are their vascular bundle patches, and these cells, they are known as medullary cells. These parenchyma cells, they are known as medullary cells, medullary rays. And the cells of medullary rays, they are changed to complete this cambium ring, to forming this interfascicular cambium. So which of the following statement is not true for the stomatal aperture? Which of the following statement? You need to select here incorrect option. So which of the statement? Which of the statements are incorrect here? I mean, they is not true about stomatal apparatus. Inner walls of the guard cells are thick. That is true. Guard cells invariably possess chloroplast and mitochondria. It's also true. Then guard cells are always surrounded by subsidiary cells. No, that's incorrect. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size, and they are known as subsidiary cells or accessory cells. Okay, so which is not true for stomata are involved in gaseous exchange. So all of these are true except C. Then meristematic tissue responsible for increasing the girth of the tree trunk is lateral meristem. It is lateral meristem, appears in the late stage and responsible for increase in girth. You are given a very old piece of Dicot Stem and Dicot Root. Which of the following anatomical structure will you use to distinguish between these two? You need to distinguish between Dicot Stem and Dicot Root piece. So which of these you will use? You will use protoxylem, presence of protoxylem or not. Then tracheids differ from the tracheary elements in tracheids differs from the tracheary elements being imperforated. Tracheids are imperforated. Tracheids are perforated, and from the other tracheary element they are differ, that is they have pores. Transmission tissue is characteristic feature of what? Transmission tissue is characteristic feature of what? Solid style. Then a major characteristic of monocot root is presence of, what is a major characteristic of monocot root? Presence of vascular bundle without cambium. Presence of vascular bundle without cambium. Then the ring girdle. So girdling experiment, that is in a ring girdle plant, the root dies first, the shoot dies together, root and shoot, they both of them dies. So which of these is correct? In that, root dies first because root will not get food in that case. So these are your homework questions, guys. So please note down all your homework questions. So I'll be waiting for your answers in comment section. So these are your homework questions. First, maximum growth in root occurs where? Second question, which of the following is well differentiated plant tissue? Which of the following is the primary meristem? Fourth, xylem in angiosperm consists of how many types of elements? Then embryo of seed is made up of what? I'll be waiting for your answers in comment section, guys. So again, let me remind you that we have a Telegram channel also, so you can join us on Telegram link also, which is given in the description box, or you can join from this link. So you will not miss out any of the live session, any of the link. We can sometimes we also share some materials, some handwritten notes also in Telegram channel. So you can follow us. You can join our Telegram link also. These are our results of the MCET 2021. You can see 95% results. 95% of them are qualified and selected. Spark Academy NEET 2021 results. You can see in 2021 97% result. Okay, our highest score was 608, and in 2021 our 19 students they got admission in MBBS, BDS or BHMS. So if you like the video, don't forget to like, share and subscribe to our channel so you'll not miss out any of the live session of biology, of maths, of chemistry, for all the subjects. So we are providing free sessions on YouTube. So subscribe to our channel and don't forget to press the bell icon for notification. Thank you so much. Like, share and comment. I'll be waiting for your comments, for your answers in comment section. Thank you so much all of you.